In this video, we are going to explore the process known as meiosis and understand how it leads to the formation of special cells called gametes, which are also referred to as sex cells. These include sperm cells in males and egg cells in females, and they are essential for sexual reproduction. The most important feature of these gametes is that they only contain half the amount of genetic material that is present in a normal body cell, and because of this we describe them as haploid cells. When two haploid gametes, such as a sperm cell and an egg cell, come together during fertilization, they form a normal cell known as a diploid cell, which then has a full set of genetic information, half coming from the mother and half from the father. This diploid cell has the ability to grow and develop into a completely new organism. But to produce these gametes, a body cell must undergo a very specific type of cell division called meiosis, which is made up of multiple carefully ordered stages. Before we look at the steps of meiosis, it helps to briefly recap the structure and purpose of chromosomes so there is no confusion. Inside the nucleus of each human cell, you would find structures called chromosomes, which contain the genetic instructions for the body. Humans have 23 different types of chromosomes in nearly all their cells, and for each type there are 2 copies, one inherited from the mother and the other inherited from the father. This gives us a total of 46 chromosomes in each normal human cell. The maternal chromosomes come from the egg and are shown in red, while the paternal chromosomes come from the sperm and are shown in blue. So when we talk about having 2 copies of each chromosome, we are really talking about chromosome pairs. In order to begin meiosis, the cell first duplicates all of its DNA so that each chromosome has an exact copy of itself attached to it, forming an X-shaped structure. Each of these structures consists of 2 arms or chromatids, which are identical to each other. After DNA replication, the next step involves lining up all of these X-shaped chromosomes in pairs at the center of the cell. Each pair consists of 1 chromosome from the mother and 1 from the father. However, the orientation of the chromosomes within each pair is completely random. This means that in some cells the maternal chromosome might be on the left and the paternal one on the right, and in other cells the arrangement could be the opposite. This randomness in chromosome orientation becomes significant during the first cell division in meiosis. In this stage, the paired chromosomes are pulled apart by spindle fibers and the cell divides into 2. Because the orientation was random, each of these new cells receives a different combination of maternal and paternal chromosomes, making their DNA different. Next comes the second cell division, in which the chromosomes within each of the 2 cells line up again at the center, but this time it is the arms or chromatids of each chromosome that are separated and pulled to opposite sides of the cell. Once this happens, each of the 2 cells splits again, resulting in a total of 4 new cells. Even though these cells might look similar, they are each genetically unique due to the mixing and shuffling of genetic material that happened during the previous stages of meiosis. These 4 genetically distinct cells are known as gametes, and in reality each of them would contain 23 chromosomes instead of the simplified example of 4 used in illustrations. In male animals, these gametes develop into sperm cells, and in female animals they develop into egg cells. If one of the sperm cells successfully fertilizes one of the egg cells, the result is a diploid cell with a full set of chromosomes. This new diploid cell then divides repeatedly through another process called mitosis, eventually forming an embryo, which can grow into a fetus, and finally into a fully developed living organism with cells that are once again diploid and capable of continuing the cycle of life.